Welcome to the Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump in, please take a moment to hit that like button, subscribe to the channel, and maybe share this deep dive with your friends and family. It really does support the Science Experience and helps us keep exploring all this amazing science. Okay, so today we're doing a deep dive into something pretty revolutionary, I think, from nature cell biology. We've all heard about mitochondria, the powerhouses, and we know amino acids build muscle over time. But what if one single really common amino acid didn't just slowly build tissue, but acted like a super fast switch, like instantly remodeling your whole metabolic engine?
That's basically the core finding here. We're looking at how a branched-chain amino acid, specifically leucine, can fundamentally and rapidly regulate the machinery that keeps mitochondrial proteins in balance. That process is called proteostasis, and this is happening right at the mitochondrial surface.
Right, the outer membrane.
Exactly. For years, we've sort of tracked how mitochondria adapt slowly, maybe to exercise or starvation, by changing their structure over days or weeks. But the big question was, how does a cell sense, immediate nutrient availability? Like, suddenly you have plenty of leucine right now. How does that translate into an instant structural change?
And this research seems to pinpoint a really precise and actually evolutionarily conserved answer.
Yeah.
Leucine itself is the direct fast response signal.
That's it. So our mission for you, the listener, is really to unpack this whole cascade. They call it the leucine GCN2 cell-L axis. We need to understand, who the players are, how they interact, and maybe why this instant signaling pathway controls so much, from your basic respiratory capacity right up to potentially disease resistance.
Okay, sounds good. Let's start unpacking that right at the boundary line, the outer mitochondrial membrane, or OMM. We often focus on what's inside the mitochondria, but this OMM is really the crucial interface, isn't it?
Oh, absolutely. Think of it like the border control for the cell's energy factory. It separates the mitochondrion from the rest of the cell, the cytosol. And the proteins embedded in that membrane are critical. They form things like the TOM complex. That's the main gateway for importing almost all the other proteins the mitochondria needs. They also handle metabolite transport, help mitochondria fuse together or split apart. If this border patrol is messed up, the cell's whole energy system starts to crumble.
Right, and because these OMM proteins are so vital, they're under constant watch, like continuous surveillance. They're constantly being tagged for removal and recycled by the cell's cleanup crew, Ubiquitin Proteasome system, or UPS. This constant turnover, this proteostasis, it's essential for keeping things running smoothly. And the new finding here is that this quality control isn't just like a baseline hum. It actually responds really dynamically to metabolic signals.
Yeah, and to actually see this responsiveness, the researchers had to get, pretty clever. They developed this novel tool in the model organism C. elegans, the little worm. They called it the mitoUFD reporter system.
This is where the experimental setup gets really cool. It's like they installed a tiny camera, right? But one that only glows green if the recycling crew, the UPS, is sort of slacking off. They tagged a normally short-lived OMM protein with GFP, green fluorescent protein. So usually, if the UPS is working properly, it chews up that protein in the GFP really fast, so you barely see any green light.
Exactly. But if that protein, let's call it the trash, gets stabilized because the degradation pathway is slowed down or impaired, then the GFP hangs around longer, it accumulates, and the mitochondrial membrane just lights up bright green.
Wow.
So it gave them this real-time visual readout of OMM quality control happening inside a living cell. They could literally watch how it responded to different conditions.
Okay, so they have this elegant surveillance system. And when they used it to screen different metabolic pathways, looking for what actually regulates that surveillance, the results were, well, you said surprising.
Genuinely surprising, yeah. They weren't just finding effects from, general protein synthesis pathways. They found that targeting genes involved specifically in amino acid metabolism had this broad, really significant impact on how quickly those OMM proteins were getting degraded.
And that led them straight to
the star player, the branched-chain amino acid leucine, or Leu. And this is where it gets really interesting because the effect was incredibly selective.
Okay, how selective? Like, could they just throw in any BCAA, those branched-chain amino acids, and see the same thing?
Nope, absolutely not. They found that giving the worms excess leucine rapidly stabilized those OMM proteins, making that reporter glow brighter. But, and this is crucial, giving them high levels of the other two main BCAAs, isoleucine or valine, had basically no effect on the OMM proteome.
Oh,
So this
Lord.
isn't just a general, oh, there's lots of nutrients around effect. It's a highly specific, acute regulatory signal being sent by leucine itself.
Okay, so if leucine is the signal, the rapid-fire message, what's the receiver? What actually senses that leucine is abundant and flicks the switch inside the cell?
The paper points directly to an amino acid sensor called GCN2. It's a pretty well-known kinase that hangs out in the cytosol.
Right, GCN2 usually sounds the alarm when amino acids are scarce, doesn't it? It tries to shut down protein production to save resources.
Exactly. That's its typical job, scarcity alarm. But what they discovered in this pathway is that high leucine levels do the complete opposite. They actually inhibit GCN2 activity. Leucine basically tells the alarm system to shush.
Wow, okay, so it's completely flipping GCN2's usual role. It's acting like a we're full stand-down signal instead of emergency conserve.
And they confirmed it too. When they experimentally knocked down GCN2, just got rid of it, the effect was identical to adding extra leucine.
Okay.
In both scenarios, those OMM proteins got stabilized because the alarm system that would normally trigger pathways to slow things down was effectively silenced.
Right. But how does silencing GCN2 inside the cell, in the cytosol, lead to proteins being stabilized on the outside of the mitochondria, the OMM? There's still a missing link there.
Good point. So leucine silences the GCN2 alarm, but who's the actual demolition crew on the mitochondria itself that gets called off?
This is where cell 1L comes in, right?
Yeah.
An E3 ubiquitin ligase cofactor. That sounds like the executioner.
That's the one, SEL1 in the worms, L in humans. It doesn't act alone, though. It typically works as part of a complex with another enzyme called HRD1. And this L HRD1 complex is the key bit of machinery that actually tags specific OMM proteins with ubiquitin, marking them for destruction by the UPS.
Ah, okay, so the pathway is becoming clearer now.
Right. Leucine comes in, high levels inhibit GCN2 in the cytosol. That GCN2 suppression then leads to less L being present or active right at the OMM. Fewer active L HRD1 complexes means less tagging of those OMM proteins for disposal. The degradation pathway gets throttled back.
And the OMM proteins are instantly stabilized. They just stick around longer.
Exactly. It's a rapid, direct line of communication from nutrient status to membrane protein stability.
And you mentioned this isn't just some weird thing happening in worms.
Absolutely not. They showed this entire regulatory mechanism, leucine inhibiting GCN2, leading to reduced cell 1L activity at the OMM, causing protein stabilization as evolutionarily conserved. They replicated the key findings, the stabilization effect, in human HEK293 cells too. So this seems to be a really fundamental piece of cell biology we're uncovering.
Okay, so we've stabilized the border patrol. What are the actual functional consequences? If we instantly stop degrading these OMM proteins, what's the payoff for the mitochondria, for the cell's energy factory?
The big payoff is a rapid overall expansion of the mitochondrial proteome. We're talking about more key functional proteins sticking around, things involved in the TCA cycle, the respiratory chain itself, transport proteins. The whole energy-generating toolkit gets beefed up.
I mean, which parts of that border patrol machinery being stabilized give the most bang for the buck, functionally speaking?
Well, they specifically highlighted components of the really crucial protein import machinery, especially a protein called TOM-M40.
Ah, the main gateway.
Exactly. You can think of TOM-M40 as like the biggest revolving door into the mitochondria. Almost every protein destined for the inside has to pass through it. So, by reducing the degradation of this main gateway complex, the cell instantly increases its capacity to import all the other proteins needed to build up its internal machinery.
And crucially, this is happening because degradation slowed down, right? Not because the cell is suddenly making tons of new TOM-M40 protein.
That's a key point. This remodeling is post-translational. It's happening because the existing proteins aren't being thrown out as quickly. The cell doesn't have to wait to synthesize new stuff. It gets an immediate upgrade using what's already there.
But that speed seems critical. Stabilizing the import gateway gives mitochondria immediate extra capacity, which then translates directly into a metabolic payoff.
A massive one. A significant acute boost in mitochondrial respiration. They directly measured a sharp increase in the rate of oxygen consumption in both the worms and the human cells just by treating them with leucine.
Wow, more power, instantly.
Pretty much. And they really nailed the causality with reversal experiments. If they blocked that TOM-M40 import machinery using an inhibitor, or if they artificially sped up degradation again, for instance, by overexpressing GCN2, that leucine-induced respiratory boost just vanished.
Okay.
It completely confirmed that the functional enhancement, the extra power, is absolutely dependent on stabilizing that OMM machinery through the specific LUU-GCN2 cell-ONL pathway.
This feels like it moves way beyond just basic cell biology. There must be implications for health and disease here. Let's maybe start with something fundamental like fertility.
Yeah, the study touches on that. It suggests that having this OMM proteostasis system working optimally is really important for, well, reproductive health. They looked at C. elegans that had a specific genetic mutation, BCAT1 E279K. Now, this mutation messes up the breakdown of BCAA, so leucine and the others build up to high levels inside the worms. It actually corresponds to a known human mutation in a gene called BCA2.
Okay, so these worms are already kind of overloaded with BCAAs because of their genetics.
Exactly, they're in a sensitized state. And when the researchers then also disrupted this OMM proteostasis pathway in these already BCAA-rich worms, they saw a huge drop in fertility, a really significant reduction in the number of offspring produced.
Mhm.
It shows how, the system is maybe a bit fragile. The combined stress of having poor BCAA clearance and a compromised OMM quality control pathway really hits organismal fitness hard.
That makes sense. Okay, and what about the flip side? What about diseases where cells might actually benefit from having hyperactive mitochondria, like cancer? BCAA metabolism is often altered in tumors, right?
It is, yeah. Tumors often rewire their metabolism, and BCAAs play a role. So they looked at human lung cancer cells, some aggressive lines like H2030 and H1666. Turns out these cancer cells naturally had higher levels of intracellular BCAAs, including leucine. And, consistent with the mechanism, their OMM proteins were already more stabilized compared to normal cells. It's like leucine was already pushing the accelerator for them.
So that stabilization mechanism we've been talking about is sort of pre-activated, giving those cancer cells an advantage.
Precisely. And here's the potential clinical kicker. These BCAA-rich cancer cells, with their already stabilized OMM, were significantly more resistant to drugs designed to shut down mitochondria, specifically drugs like MB12 that inhibit protein import.
Wow.
The stabilization effect, driven by leucine, seems to give these cancer cells a survival edge. It keeps their metabolic engines primed and makes them tougher to kill when you try to target their mitochondria.
That's a really powerful connection. The amount of leucine floating around in the cell cytosol is basically talking directly and instantly to the mitochondrial surface via this GCN2 cell 1L axis. And it's dictating the mitochondrion's capacity, its resilience, its ability to function.
It really reframes how we should think about leucine, doesn't it? It's not just a slow-acting building block that you incorporate into muscle over days or weeks.
No, it's acting like an immediate metabolic thermostat, or maybe a rheostat, fine-tuning the power grid.
Yeah, a rapid signaling molecule.
Okay, so here's the final provocative thought I want to leave you, the listener, with. This discovery really changes the game for leucine. It's acting as this rapid, conserved signal that dictates mitochondrial capacity almost instantly. If the cell's entire energy engine can be structurally and functionally remodeled within hours just by changing leucine availability, as this research strongly suggests, what does that really imply about the acute, immediate impact of our dietary choices, or maybe even supplementation, on our fundamental metabolic health?
It's a fascinating question. The idea that we could potentially fine-tune our core metabolic machinery almost instantaneously just by tweaking external nutrient signals like leucine, well, that opens up a whole new frontier, doesn't it?
A truly incredible deep dive into the real nuts and bolts of our cellular engines. Thank you for guiding us through this really cutting-edge science.
It was my pleasure. Really interesting stuff.
And thank you, the listener, for joining us on this deep dive. Please don't forget to like, subscribe, and share this with anyone else who gets excited about science. Remember, your support is what makes the Sans Science experience possible. We'll catch you on the next one.